glaube, das halten Sie erstmal.
And we come back to a topic that was stressed already at the beginning of the session. And now Marko Loparic will give a talk, and it's about nanomechanical profiling of breast cancer. So I think we are back to the medicine.
Okay, thank you.
Marko, the floor is yours.
Thank you, Bert, and thanks for staying so long, in this very challenging afternoon, right, to keep the attention. I will try to be short, precise, and, enlightening. So, as Bert said, we have today already Philip Oertler presentation where you saw the basics of the AFM, the potential, the benefits of AFM in the research, and actually what I will present you now, it's a big jump in the evolution of the AFM. Actually, it's the first AFM built to become medical device, right? And it's called ARTIDIS, which stands for Automated and Reliable Tissue Diagnostics. And our first actually application, which we found of particular interest, is breast cancer diagnostics and prognostic. Why breast cancer diagnostic and prognostic? You can see on the following slide. So 1.3 million overdiagnosed and overtreated for breast cancer over the past 30 years. False positive mammograms, 2.8 billion annually. Breast cancer overdiagnosis is 1.2 billion. Incident rate doubled. So this is an emerging problem. This is the problem which doesn't have the good solution. And this is the problem actually of the overdiagnosis. And when we talk about the overdiagnosis and the overtreatment, then we are talking actually about the chemotherapy. And I think everybody is aware of the profound side effects of the chemotherapy. And this is something what absolutely has to be resolved, because there is the many women, as I will show you later, they don't have a benefit of this chemotherapy, right? And ARTIDIS is our proposition how to resolve this issue. So why we have this problem today? Why clinicians cannot resolve it? Simply because we don't have reliable markers which will help them, which they can use to get information or to get exact information about the disease progression and the recurrence. And clinicians are doers. So when something is not clear, they will rather do everything what is possible, not maybe the best what is possible, because maybe there is nothing better. And therefore, we have such a high amount of the overdiagnosis. ARTIDIS is the proposed solution to this problem, and ARTIDIS, in the simple words, as you saw on the first lecture, actually, is a very simple method. It came relatively lately because of the technological advancement which are necessary, which are required, and one of these is definitely the tip, which you can see here, which is very sharp, so it's only 50 nanometer. In diameter, and this is about 4,000 times smaller than the hair shaft. Why for only 50 nanometer? Because this is the resolution which we need, right? And you see on the rest of the picture, actually, it's a cancer cell, and this is the cancer tissue. So the only thing which is fake on this picture is a color, right? This is the real picture of the cancer and the tip. And ARTIDIS actually is a means measurement of mechanical properties, or in simple words, stiffness of breast tissue samples at molecular level. Now, why this is so important, right? As you can see on here, on this pretty stuff slide, a lot of attention today we have on genes, right? We have next generation gene sequencing, we have molecular profiling, which is also connected with the gene expression. And in principle, this is very important, but also on the other side is very complex, right? Amount of genes which are involved in the cancer progression, in the cancer development and progression is enormous. And therefore today, you have very cheap genomic tests, for a few €100, but then analysis costs probably like 100,000 euros, right, of the same results. And this is the big problem because they are very complex. So we took completely other side approach. We simply ask why cancer cell can migrate, right? Which properties they have that they can migrate, because normal cells, we don't find the brain cells in the stomach and vice versa. And cancer cell has the ability to migrate. So we simply ask the question, what are these, what are their characteristics? And actually, the other good part is that, yes, they have certain characteristics that they can invade. As you can see here, in the first phase of the cancer development, they break the basal membrane, and then they're migrating, or they're progressing through the extracellular matrix to the blood vessels, and then they go through the body. And it's absolutely true that this migration and this phenotype is result of many biological pathways involved, right? But we have here, much simpler job to do, actually. Instead of looking for all the pathways, all the genomes, all the genes which are involved, we are simply measuring the final effect. We are simply measuring how cells are fit, to do metastasis. And in the healthcare or in the diagnostic ecosystem, actually, we are located, on the phenotype side. For most of omics, which are today, the most modern approach in a diagnostic. So our method actually is placed on the phenotype side, somewhere where is pathohistology. The difference to the pathohistology is that we are much faster, as you will see later, and we are working with the fresh tissues, right? So for atomic force microscope, for RTDs, there is no need for any processing. And how practically, actually, now we are doing the measurement. So we are in vitro medical device. Therefore, we are analyzing the biopsies, which are extracted using core needle and with a navigation from the ultrasound. So once this biopsy is extracted, this biopsy is straight away analyzed. So we don't have to have any chemical, mechanical processing, right? This biopsy we place under our machine, and then we are measuring across the sample. And we are simply doing indentation testing for 10, 15,000 times, right? And for each in this indentation cycle, we are calculating the stiffness. Finally, out of these many indentation cycles, we are plotting histogram where we can see actually the mechanical profile. So we see which stiffnesses are occurring more frequently, right? And as I said, very important for our device is that we are working with the fresh tissues, and we don't affect the tissue during the measurements. That means that after we are done with our measurements, the tissue can be returned back to the histopathology and can be used for all analysis. So therefore, we don't need extra tissue. We are 100% compatible with the existing clinical cycle. And our biggest breakthrough so far is shown actually on this slide, where we simply measure the real patients, right, in a clinical study, and by measuring which were extracted from these patients, we come to the fascinating result that we can distinguish healthy, benign, and cancer with a high statistical power. And this you can see here. And this paper was published in the Nature Nanotechnology back in 2012. Today, we are proud to show that we have also a research prototype, which is now, let's say, state-of-the-art AFM for measuring of the stiffness in the living tissue. And with this device, actually, we reached this proof of the concept level. In the same time, the device is used now in the hospital, is used in the 2 ongoing clinical trials, and this device we will use as a basis for final medical device development, where, of course, there are regulatory and standards and frames which have to be actually developed. The first use case which we are proposing is called Arthritis Diagnostic. And Arthritis Diagnostic, it's actually asking simple question: do I have a breast cancer? And today we have histology or histopathology as the standard way how the diagnostic is working. And as you can see here, each of these blocks is one process, right? So this is heavily complex system. There is an extensive infrastructure behind these processes. So the pathology lab, they have multiple machines. They have 4 to 5 specialists actually who are involved in the whole process, and it's pretty lengthy. So it's between in the best countries, in the best labs is 2 days, but usually it's up to the 7 days, right? On the other side, we have Artidis, which is now confirmed on more than 200 patient samples, and where we have very clear distinct parameter between the healthy cancer and benign. And the best part is that we can do this measurement in only 3 hours, right? And as I said, this we also confirm if you want to see more details from the especially technical part. So, for example, which cantilevers we use, right, and which setup you can take a Nature Nano, and there we have more details. From the point of the medical device, which we are proposing to be used in the clinics, the significant improvements are coming from the faster analysis and results delivery. This is at 3 hours instead of days. We have simple, straightforward analysis. As you saw, this prototype is the device, which doesn't require a big room, big infrastructure. It can be operated by the average user, meaning there is no need for a high specialist, right? And because of the whole design and construction, it's much more affordable comparing to the standard pathology. The message which we want to achieve here is that after only 3 hours, 70% of patients analyzed by Artidis Diagnostic will go home contented or happy. Why 70%? Because from all the biopsies which are collected, 70% of the biopsies are benign. Benign means that those patients have a very excellent prognosis, and in principle there is absolutely no need today that they are waiting up to 1 week, that they experience all the anxiety and all the stress in waiting for results. Therefore, for 70% of the patients, analysis by Artidis Diagnostic could be substantial improvement, right? So in principle, they come to take the biopsy, biopsy is analyzed, after 3 hours there are results, they can go home, that's it. There is no need for further diagnostic. For other 30%, which are actually cancer patients, there is also a big benefit. Why? Because now they can plan further, right? There is no need for further delay. It can be immediately consulted, and as you will see with a further use case, in 3 hours we can get actually much more than just diagnostic. We can also get the prognostic factor. We can get the information about the aggressiveness of the cancer. And practically 70% of the patients, that means about 1 million patients only in US annually, right? So we are talking about a large population which will benefit from those results. And second big actually issue, this is how I started this presentation, the problem of the over-treatment and over-diagnosis. The big question today in the breast cancer treatment is, do I need the chemotherapy, right? You've probably heard for tamoxifen, for Herceptin, for estrogen positive types of the cancer which are successfully actually cured, but there is still always question, in addition to this hormone therapy, do we have to add a chemotherapy, right? And the chemotherapy, as I showed in the beginning, has some substantial side effects. So, do I need a chemotherapy? This is the slide which is actually very showing the whole problem today. Based on this 10-year survey, they show actually that the absolute benefit from the chemotherapy, in addition to the hormone therapy, which is a standard, is only 4%. And therefore, it's clear that over too many patients are getting chemotherapy today. And what we found that with Artidis, actually we have substantially higher sensitivity and specificity in terms of recognizing the aggressive cancers, because we can easily select the patients just by measuring the healthy tissue which is surrounding the cancer, right? And which is declared by the histology, this is a healthy tissue. There we can actually analyze this part of the tissue and see which patients actually are highly aggressive and which are low aggressive cancers. And this is something which also we find a correlation in metastasis. So in principle, when we measure this healthy part and we find that this healthy part is not healthy, this is the sign that the cancer is spread, and this is the highly aggressive cancer. For Artidis Prognostic, we have now ongoing clinical trials as well. We are expecting soon results which will cover the 5-year follow-up period. And the part which we want to achieve is that with Artidis Prognostic, more breast cancer patients will maintain the quality of their life, because we will help oncologists to select the patients which will really benefit from the chemotherapy and which wouldn't benefit of chemotherapy. This is just shortly to show our network. So our device is already used in a few centers, and we have already a lot of clinicians on board who will use our technology. Nuomedis was found 2 years ago, so we are still a relatively young company. It's actually results of 10 plus years of the basic research in the University of Basel in the Biozentrum. We have currently 7 people, and we have our strategic collaborators in the University Hospital Basel and the Biozentrum, where you have mixture of clinician, physicists, biologists, and so on. We have IP protection, and our main patents are granted in US, Europe, and Asia, so we can say we have worldwide protection. We have these 2 clinical trials, and I will also use this opportunity to announce that we are currently in the investment round, so we are going for the next capital increase, and this will be exactly to accelerate the development, that we can enter the market fast, and that we can actually enable clinicians and the patient to use the benefits of the device. This is how the final result should look like: very small portable machine with disposable holders, and that's everything from me on this lecture.
So Marko, thank you very much. I think we have only time for one very fast question. Okay, Barbara.
Can you also use it for other cancer types?
Yes, of course, this I didn't mention because of the time restriction. We have already feasibility study on the prostate cancer, colorectal cancer, lung cancer, and a liver cancer. Well, it's not the same, but we show that as well in these cancers we have a very strong difference between the healthy benign and in principle our goal is to bring to get a certificate for the TDIS, that it's a clinical or it's a medical device, and then of course we will use it for broad application in clinics. In addition, just one more, maybe sentence, in addition we have collaboration with the pharma companies, because today the big problem with pharma companies are biomarkers. So they have a lot of new drugs, and they don't know exactly which patient will benefit. This is exactly something what we also have the active collaboration. Already some publications will occur now.
So thank you, the speaker, again, and
200.
I thank all the participants for discussions and so forth, and for staying such a long time with me. So coffee time.